This is Research Like a Pro, episode 246, RLP with DNA, Chapter 8, Part 2, Segment Data Tools. Welcome to Research Like a Pro, a genealogy podcast about taking your research to the next level, hosted by Nicole Dyer and Diana Elder, accredited genealogist professional. Diana and Nicole are the mother-daughter team at FamilyLocket.com and the authors of Research Like a Pro, a genealogist guide. With Robin Worthland, they also co-authored the companion volume, Research Like a Pro with DNA. Join Diana and Nicole as they discuss how to stay organized, make progress in their research, and solve difficult cases. Let's go! Hi everyone, and welcome to Research Like a Pro. Today's episode is brought to you by findagrave.com, the best place to search online for burial information for your family, friends, and famous people. At Find a Grave, you'll find details about cemeteries and individual memorials for the people buried in those cemeteries. Hi, everyone, and welcome to Research Like a Pro.
Hi, Nicole. How are you today?
Great. Just been using Airtable a lot and loving it. How about you?
I've been doing the same. I'm working on our study group project, and it was the timeline assignment. And I'm working on proving through DNA my connection between Thomas Beverly Royston and his father, John Kerry Royston. I have a really mammoth research log in Google Sheets that I use for accreditation, back 2016. It's got like over 150 rows. And the problem with those 150 rows, I can't group and sort it like I like to do on Airtable. So I imported the whole thing into Airtable, and I am so excited to have it there and be able to manipulate it, to look at all the different people and all the different records. It's a little bit of work to put it in there, but now it's done and I'm ready to use it. It's pretty exciting.
Awesome. So you can sort in Excel, right? But you just can't really group?
Yes. So I did it in Google Sheets, and I would sort it by record type, or I would sort it by location. I think those were the two main things. I had set up the columns for sorting, but I couldn't do the grouping, and I couldn't create a new view, say, of just John Kerry Royston, because for that 4-generation project, I had to detail all of his wives, all of his children, and he had at least 2 wives and children by both of them. So when I say research log, it is really thorough of his entire family. And often I want to just look at like one of his sons or look at one of the wives. So I had to add some fields when I did import it into Airtable. I had to add an individual field so that I could easily group by the person or do select views. I guess for selecting views, I could have maybe just any mention of this person. But anyway, I added a couple things so I could really use it how I wanted to.
How great. I was just using Airtable for a project with managing different course members in our online courses and just, working on that. And I was trying to use an Excel CSV file, and I was annoyed that I couldn't filter it and group it the way I wanted to. And I thought, why am I even doing this? I'm just going to put this in Airtable. So it was so easy to import it, and Airtable recognized certain fields that could be a certain field type and just gave it that field type, and it was so nice.
That is awesome. So you just imported the whole thing without selecting any fields or...
Right.
Wow, that's awesome.
It was nice.
It's fun. We're always learning more about what we can do with Airtable. Well, let's do announcements, which brings me to we have our new Airtable Quick Reference Guide available as a PDF download from our website. And we have our Research Like a Pro webinar series for 2023 going, and we are so excited to be sharing monthly case studies featuring the Research Like a Pro process and projects that have had DNA added to them as well. Our next Research Like a Pro study group is this fall. We'll begin the end of August and end the beginning of November. And we have our peer group leader application on our website. So if you've been thinking you'd like to be a peer group leader, we invite you to apply for that. You'll get complimentary registration and get to work with a small group of people in the study group. We have our newsletter available for the latest and greatest that we are doing, and we have our coupons that come through that newsletter for our courses. And also the next big conference, now that Roots Tech is over and done, is the National Genealogical Society Conference. That's in Richmond, Virginia. That's open for registration, so you can attend in person or you can be part of the virtual segment of NGS. So I invite you to go check that out on their website and get registered for that.
Yes, and although I won't be able to attend NGS, we are going to have a booth there. So if you want to come find us in the expo hall at the NGS conference, then you can see Diana, and I'll be managing our virtual booth from home.
Right. We're excited to be there in person, and we'll have our books and we'll have flyers, and it's always just fun to talk to people. I love, hearing how your research is going and, just what's going on with fellow genealogists. It's awesome to connect.
Right. And if you want a laminated version of our Airtable reference guide at conference booths is pretty much the only place you can get it because we don't sell that on our website. We only sell the PDF download. So that's another fun reason to come see us at the booth.
That's right.
All right. Well, let's get started with today's topic. Today we're talking about Research Like a Pro with DNA, Chapter 8. And last episode, we covered the first part of the chapter about genetic networks and pedigree triangulation. And today we'll talk about the second part of the chapter, which is about segment data and chromosome browsers. So let's talk first about segment triangulation. How can this help you? Well, this method can sometimes help you trace your family tree back even more generations than you knew you could. And triangulation relies on DNA segments that are shared by you and at least two of your matches on the same chromosome who have the same segment, and it's coming from the same ancestral source. And if the common ancestor shared between these three or more individuals can be figured out by looking at family trees, then that triangulated DNA is compelling evidence supporting a proposed relationship. The way this works is that somewhere back in time, your ancestors passed DNA to their children, who passed it to their children. And over time, that segment of DNA that was passed on got a little bit smaller and a little bit smaller, and it eventually ended up with you. So the goal of looking at segments and doing triangulation is to figure out the ancestral origin of the segment that's shared between you and two other individuals. Sometimes you might have a segment of DNA that you can't identify a common ancestor, and that could be that the common ancestor is just further back in time than there is documentary evidence available to trace the trees back that far. To make sure that you're using a segment that's all shared by the same people and that it's a valid segment to analyze, the segments need to overlap enough that it's a long enough segment of DNA. So if you're only overlapping for like 3 or 4 centimorgans, that's just not quite enough. You want to have segments that are closer to 10 or more centimorgans in size. So only some of the DNA testing companies have segment data to do triangulation with. GEDmatch, MyHeritage, 23andMe, all three of these have the ability to report segment triangulation. Family tree DNA provides segment data, but they do not provide triangulation information. You have to do the extra work to contact your matches and figure out if the other two individuals share that same segment with each other. Ancestry DNA does not provide segment data at all. So you cannot do segment triangulation if all you have is Ancestry DNA, but you can transfer or upload your Ancestry DNA results to other companies to find matches there and to see the segments you share with others at other databases. So one of the key takeaways from this section about triangulation is to make sure that it's a true triangulation. You need to make sure that the other people who are part of your triangulation group share that same segment with each other. And it's not enough that you share with one person and you share with a second person at the same spot. That's only 2 legs of the triangle. The third leg of the triangle is seeing that those other 2 people also share that same segment. And you might be wondering, why wouldn't they? Sometimes that does happen where it's just on a different chromosome, so they aren't sharing that. So we just have to be careful and aware of the possibility that they may not share that same segment.
Well, thanks for taking us through that. Those are some really good things to think about because there are limitations to what we can do with our DNA, and we need to be aware of that. Well, you may be wondering how you even look at the chromosomes on the different testing companies, and as Nicole mentioned, we can't look at it on all the companies, but there are 3 testing companies and GEDmatch that do provide a chromosome browser. And the Chromosome Browser lets you visualize the DNA that you inherited from common ancestors and lets you compare segments that you share with DNA matches. And so you'll be able to see the chromosome number, the start and end points of each segment. But an important thing to remember is that you have 2 copies of each chromosome. So you inherit one from your father and one from your mother. And if you're going to try to triangulate, like you were talking about, Nicole, you have to make sure that these overlapping segments are on the same chromosome copy. And that can be tricky because you got to compare the right chromosomes. So if you examine the DNA segments you share with your DNA matches and know how you are related and the common ancestor you share, then you can assign those segments to that ancestor, which is really exciting. And after that segment has been assigned or identified as belonging to an ancestor, then you can compare segment data from other DNA matches. Just keep in mind, just because the match shares the same segment on the same chromosome, it doesn't mean they necessarily descend from the same set of common ancestors. It could mean they descend from an ancestor of the most recent common ancestors. So, for example, I share a lot of DNA with my first cousin. We also share a lot of great-grandparents and great-great-grandparents and great-great-great-grandparents. And so we have to think about that little segment of coming back, much further perhaps than we would initially have thought of. So it's really fun to start working with this, and the more you do it, the more you get comfortable with it. So if you've never looked at your Chromosome Browser on your DNA testing company, here's where you can. MyHeritage has the Chromosome Browser one-to-many. 23andMe has Advanced DNA Comparison. Family Tree DNA has Chromosome Browser. GEDmatch has the tool titled One-to-One Autosomal DNA Comparison or One-to-One XDNA Comparison. And then for their Tier 1, which is their paid level, they have the 2D Chromosome Browser, the 3D Chromosome Browser, and Compact Segment Mapper. So you'll see that GEDmatch has quite a few tools that you can use. So I'd encourage you to just go log into your DNA and take a look at those chromosome browsers and see what you can find and understand what's going on with that reporting of your DNA.
So, as I was talking earlier about segment triangulation, I was thinking about how in the past, segment triangulation has been talked about as the gold standard for genetic genealogy. And I think a lot of people have the misconception that you need to have segment triangulation in order to write a proof argument. And that's just not the case. In recent years, clustering and pedigree triangulation has kind of become more of a tool that's used more often. And using the total amounts of shared DNA with matches and not including segment data is an acceptable way of writing a proof argument. And you can look in various case studies in the National Genealogical Society Quarterly and see that not all of them do segments. Where there is segment data available, it can add additional evidence to the body of evidence. But where it's not available or where it's not necessary, it's not used. So I don't think you should take away from this that you have to use segment triangulation in order to feel that you have reached the level of proof. It's a tool, just like the other tools, and it can definitely be useful, but it's not always required.
Right. The genealogy world has changed a bit with their interpretation of how to use the segments, which is, I think, a good thing because of that random recombination. We're just not going to inherit as much. It gets so much more difficult to try to triangulate on a segment when we're working far back in time. It's much easier to gather this body of evidence of shared matches and all these people in the same genetic network. So it's a good shift. But I think it's just so important to still understand how to use segments, which is why we're talking about it today.
Right. And it can be so helpful to find more matches that are relevant to your case. And so the next thing I'm going to talk about are some third-party tools that can help you with that to identify DNA segments that are relevant to your project. So let's go over some of these segment triangulation tools. Genetic Affairs at geneticaffairs.com, they have a tool called AutoSegment. AutoSegment is able to be used for segment data from MyHeritage, 23andMe, Family Tree DNA, and GEDmatch. What's great about this is that it can take matches and segments from all these websites and put them into one report. That's really useful because it's kind of helping you compare segments across companies. And You can't do that with just shared matches, but when you have a specific segment that people are sharing, you can see if that segment is shared by somebody at 23andMe and somebody at MyHeritage. There's another tool at Genetic Affairs which is very similar, and it's called the Hybrid Auto Segment tool. This is the one that puts all of the data into one report. The regular auto segment tool just puts together a report from each company separately. So the hybrid auto segment tool is the one that's pretty exciting. And having all that segment information in one place can really streamline the process of analysis. That can just be a huge time saver. However, I will say that when I've used that in the past, it helps to limit the parameters so that you don't have such a huge report or a massive spreadsheet, because for one, it can become too large that you can't load it. And sometimes it doesn't include all of the people that you wanted to include because it was too large. So just be aware of the limitations of that. Another option for compiling DNA segment data is to use the DNA GEDCOM client, and that can help you extract data from testing companies. And then you can use different tools like JWorks and KWorks, which are tools from DNA GEDCOM, and then look at that data in a spreadsheet. So then you can see how the DNA segments that you share in common with your matches are forming triangulated groups. And then you can work on identifying the most recent common ancestor. So to use DNA GEDCOM, you just need to get an account there, and you need to subscribe to get access to their program called the DNA GEDCOM client. Then using the client on your computer, you can download your files from Family Tree DNA, 23andMe, MyHeritage, and GEDmatch. Downloading the files does take some time, so you might need to set your computer to not go to sleep and then let it run overnight to extract all the data. Once you've gotten all the data, then you can use the tools. And so one of the tools is JWorks, which is a downloadable Excel macro program that sorts your segment data. Another one is KWorks, which is an online version of JWorks created by Kitty Cooper, and that sorts your DNA data and creates a spreadsheet of overlapping segments. And this is accessible through the DNA GEDCOM website. If you subscribe to GEDmatch Tier 1, you can also use the Autosomal DNA Segment Analyzer at DNA GEDCOM. And this program reports which people share DNA in common with others. And so it kind of helps you go beyond just the overlapping segments to finding likely triangulation groups. All of these tools are helpful for streamlining the gathering of your data into one place and examining and analyzing it to figure out the DNA segments you inherited from specific ancestors.
Right, and I think it just takes some experimenting to see how to use it. And we always teach to research by objectives. So if you have an objective to research maybe a fourth great grandfather, it could be really helpful to try to find some segments that you and your matches all triangulate on, or just to even identify some of those chromosomes. Which leads us to our next topic, which is visualizing DNA segments. And this is one of my favorite parts about working with segments. We call it chromosome mapping, and it aims to identify the ancestral origins of specific segments of DNA. And I love visual. Anytime there's a visual, that's what I'm all about. So through this process, you can see which segments you inherited from your specific ancestors. Now you have to remember, you can't visualize your own DNA. You can only see the DNA you share with your DNA matches. So when you have a chromosome map, you are the base person, or your test taker is the base person, but we'll talk about just our own. We are the base person against which our DNA match is compared. And the chromosome map will show the segments you share with the DNA match. So you can use your autosomal DNA results to build this map of your chromosomes, and we have some great tools to do that with. The first one is DNA Painter, which is my favorite because it's easy and it's fun and it uses color and it uses just segment information. It doesn't use your raw DNA, so that ensures privacy because people can't see the nucleotides in your raw DNA. You just get to see your segment information. But once you identify a common ancestor between you and your genetic match, then you can assign that bit of DNA, that segment, to a match, and you can paint it. So whenever you hover your mouse over a specific little segment that you have painted a certain color, you can see what the names of the ancestors are. And as you continue to do this, you will eventually create a chromosome map. DNA Painter is fun because it tells you how much of your genome is painted. And I think at this moment I only have about 50% of mine painted. I need to return to that. But one of the problems is that we can't use Ancestry data, which is where so many of our matches are. And so we have to use our results at 23andMe, MyHeritage, and Family Tree DNA to paint our chromosomes. Or GEDmatch, if people have uploaded to GEDmatch their Ancestry DNA, we can use that. But it's a fun visual. And the way that it can really help you is if you have identified some DNA segments from, say, a third great grandfather because you have connected with that DNA match, you know what your common ancestor is, and you've done the tree work to see that their tree and your tree all go back to the same person, then if you get a new match who also is on that segment, that gives you some clues about how that match fits into your family tree or who your common ancestor could be. So we can really use it for some good analysis of our DNA and to give us some help. So one thing to know, when you go to DNA Painter, you can create your chromosome map for free on DNA Painter. And when you go to create it, you will see 2 lines for each chromosome because we have a maternal and a paternal chromosome. And on DNA Painter, the top line represents the paternal copy and the bottom line represents the maternal copy. Now, that is not always the same with the other websites. You can't always assume that it's paternal and then maternal. So just be careful with that, but know with DNA Painter, that is how it is set up. And you get the chromosomes from 1 to 22, and then you even have the X chromosome at the bottom of the map. So it also features a cluster auto painter tool, which gives you a new chromosome map from the DNA segment cluster data in cluster HTML files and auto cluster reports from some of those companies we've been talking about: Genetic Affairs, MyHeritage, GEDmatch, and the Colin Leeds method at DNA GEDCOM. So a lot of really neat things that you can do with DNA Painter. Now, there is one other tool that you could experiment with, and that is GDAT, so GDAT, which stands for Genealogical DNA Analysis Tool, and it's an app for Windows, Mac, or Linux that helps you use your autosomal DNA test results for genealogy. So you may have used back in the day the tool Genome Mate Pro, and this is a replacement for that. Unlike DNA Painter where it's all online, this one you use on your own computer. And so some of the things it can do are chromosome mapping, triangulation, in common with grouping, the ability to mark most recent common ancestors, XDNA, inheritance path ancestors. And so it's something that if you are willing to spend the time and learn a new tool, you might love it. It just depends on how your brain works and if it's something that clicks with you.
Right? That could be said for all these tools because some of them I just never use and other tools I use all the time. So we kind of develop our favorite tool and it helps us, we get good at it. It helps us find the matches that are relevant. And I think that's okay.
I do too. Sometimes it's good to experiment with new tools and see if we might like to use those for a certain project or a case. But if we get really good at some of the tools, then maybe that's just fine. You know, we don't have to try every single tool that's out there.
Right. I usually focus on my network graphs and figuring out groupings of people that way. And then, like with one of my proof arguments recently, after I had come to my conclusion, I wanted to add some segment data. So I just kind of went to GEDmatch and I knew there were certain people that I'm already using in my project that are in GEDmatch. So I just went and kind of looked to see if I could see any triangulated segments there with some of the basic tools for segment analysis. And sure enough, I did find one. So I just created a simple table for my report, including the segment data of this triangulated segment. So I didn't use a fancy tool to help me find that. I just used my other tool to figure out my whole case. And then at the end, I used a simple triangulation tool to add a little bit more evidence.
Nice. And that was for a specific project. That's not necessarily what you would do for every single project. So I think we have to remember, especially with DNA, that different projects might warrant some different treatment of how we work with the DNA.
And now for a word from our sponsors. Findagrave.com makes it easy to find the graves of ancestors, create virtual memorials, and add photos or virtual flowers to a loved one's memorial. You can search by name, location, cemetery, date, and more. Find a Grave members add more information each day. So if you've searched the site before without finding what you're looking for, come back and check again. Looking for a way to give back to your community? Download the free Find a Grave app, visit your local cemetery, and add missing grave information to the site. It's an easy and fun way to spend some time outside and help other people who are searching for their family and friends. Find a Grave is completely free to use, so start your search today at findagrave.com. Okay, let's talk about a new topic, visual phasing. If you hear that word and it strikes fear and dread into your heart, that's because it's a more advanced and challenging tool. And that's okay. It's a time-intensive way to map your chromosomes, and it can provide a detailed map that identifies the DNA segments inherited from each of your 4 grandparents. So for that reason, it can be very beneficial. In DNA Painter, like Diana mentioned, sometimes you only have 30 to 40% painted, whereas with the visual phasing, often you can complete an entire chromosome map so that you know at each part of every chromosome which part was inherited from which side of the family. When I'm looking at my best chromosome map that I've done, by best, I mean most complete, it's my grandpa Elder's, and it's 48% complete. I thought that was pretty good, but still, chromosome 1, the paternal chromosome, is pretty empty. That's like the longest chromosome. And I barely know anything about that chromosome. I just know like 2 small segments and where they came from. So, depending on how many matches you have in the companies that have segment data, you may not be able to identify just through segment painting on DNA Painter all of the chromosomes and which parts of them came from which side of the family. So visual phasing can help you do that. But there's a lot of requirements to get started with it. One of them is that you need to have a set of 3 siblings. And so if you don't have 3 siblings, it's going to be a lot harder. And some people have tried to do it with 2, but it's just much better to have 3. So I don't know that I would recommend trying it unless you have 3 full siblings. And it does take focus and patience. Karen Stanberry describes it as a logic puzzle, like Sudoku, where you're sitting down and you're trying to figure out the answer to a puzzle almost. If that sounds fun to you and you have 3 siblings, then you might want to consider trying it. What you need to do to get started is get the Excel program that Stephen Fox wrote. And so if you join the Visual Phasing Working Group on Facebook, then you can get that Excel program. There's a 5-part series on Blaine Bettinger's website with instructions on how to do visual phasing. And he also has a whole chapter in the book Advanced Genetic Genealogy, edited by Debbie Parker Wayne, that teaches about visual phasing and how to do it. And that's kind of what I've used as I've learned to do visual phasing.
Yeah, I love reading Blaine's chapter and his blog posts and attending his classes on visual phasing. I think it's just a fascinating process. But I haven't tackled it because I don't have 3 full siblings. I only have 1 half sibling. So, it hasn't been high on my list to do. But I think you definitely should keep working on that.
Honestly, I haven't felt that I need to. It's just, honestly, the number of matches on Ancestry with trees make it so that that's where I focus most of my time. Ancestry matches don't have segment data, so I spend my time on the low-hanging fruit. And like I said, sometimes I will look at segment data later, but it's not currently in my process to use a lot of segment data.
Well, and don't you think that a lot of these tools were developed, years ago, and we did not have the number of DNA matches that we have now, especially on Ancestry. And so, people were trying to figure out how to use DNA and how to figure more things out. And so they developed a lot of these tools. And so I'll be really curious to see what we are using, a year from now, 2 years from now. We may completely change our methodology using different types of tools. So it's really evolving.
Right. I would say that probably the tool that I use is DNA Painter to map segments. So
same.
I highly recommend that.
Yeah.
So there are some challenges with mapping chromosomes and DNA segment painting. And one of them is that you may share multiple common ancestors with one of your matches. So then how do you assign a DNA segment to one set of common ancestors? If you have a double cousin, then which set of ancestors did that segment come from? You just have to use caution with those and maybe wait and compare multiple matches. And then over time you might be able to figure it out. But those cases where you do have double cousins, pedigree collapse, and endogamy are actually the times when mapping DNA segments can be even more useful because it can help you sort things out and help you figure out, okay, this segment seems to have come from this side of the family, and this segment came from this other common ancestor that we share. So then it can help you use that as evidence because then you've kind of figured out how much you actually share because of a certain common ancestor. So it can be a challenge, but then also a solution to a challenge. So let's just go over really briefly some situations that you may see in your family tree that you need to be aware of as you're doing segment mapping. So I mentioned pedigree collapse. This happens when people who are related, like first or second cousins, get married or have children together, then those children have pedigree collapse because they have fewer unique ancestors because their parents are related. So their pedigree, instead of getting bigger at each generation, at a certain point, the pedigree only has 6 instead of 8 unique ancestors. Endogamy is similar, just more severe when pedigree collapse occurs over and over in someone's tree because people choose to marry within their population group. This sometimes can occur in geographically isolated populations, religious populations. The same type of effects from endogamy can occur when you have different things like founder effect or different problems where a lot of a population is wiped out and then those who are left are kind of the founders and they, marry each other. So anyway, the children of these communities with endogamy and populations that have similar effects to endogamy, the children inherit a lot of DNA from the same people. And so that can really introduce challenges for separating DNA segments into specific common ancestral couples because they may share a lot of small segments with matches, and those all came from different common ancestors because they share with the same person in 4 different ways. They're a third cousin on this side, a fourth cousin on that side, a fifth cousin once removed, and a sixth cousin. And so if they are that many relationships with one match, it just makes the analysis and the segment mapping quite challenging. And then the last challenge is that there's sometimes a lack of pedigree completeness. And so if you don't know your tree and it's not complete to a certain level, then it can make it really hard to identify where a certain segment of DNA came from. And of course, we all have brick walls or dead ends in our tree, so we can all relate to this. But if you have several lines of your family that can't get back further past a grandparent or great grandparent, then that's going to complicate your segment mapping quite a bit.
Absolutely. Well, that was a great discussion on segment mapping and visualization of our segments. So many fun things to consider and try out. So let's shift gears a little bit and let's talk about some DNA tools and methodology for the other kinds of DNA inheritance. We've been talking all about autosomal DNA, but now we'll do a little bit on mitochondrial Y-DNA and XDNA because those are very useful types of DNA inheritance that can help us to discover more about our ancestors, and we want to know what kind of tools we can use for those. So we'll start with mitochondrial DNA, and the tools that we use for mitochondrial DNA are haplogroup reports and the matching report at family tree DNA. So we get these reports, and what do we do with it? Well, just like any other source, we analyze it, we correlate it with our documentary research to try to draw some conclusions. So here's what you get from each company concerning mitochondrial DNA. Family Tree DNA is the only company that offers the full sequence mitochondrial test. So this is where you will actually get matches on your mitochondrial line. But just know that those matches can be very, very, very far back, and they don't have surnames coming through the female lines, and so it can be very difficult to figure out how you connect to them. But we use it for targeted testing. So if we find an ancestor and we find a very possible mother or a sister, we can trace down their descendancy lines and find a mitochondrial test taker that we can compare to. And so it's very useful. We just have to understand it. Now, what if you've tested at 23andMe? Well, you will get a high-level haplogroup for your mitochondrial haplogroup, and you get your autosomal DNA results. And so sometimes you can compare and see if people are on the mitochondrial lines just from their haplogroup. Living DNA also reports that haplogroup and gives you autosomal DNA results. MyHeritage and Ancestry include tested mitochondrial DNA markers in their raw data download files. And so if you want to, you could maybe somehow use that to estimate a mitochondrial DNA haplogroup. And then there are tools for estimating a mitochondrial DNA haplogroup, such as James Lick's tools at jameslicks.com. So we don't talk a lot in genetic genealogy about using mitochondrial and different tools, but it's just interesting to think that it might be a way to break through a brick wall in one of your female ancestors. One of the things you want to do is you want to actually see which ancestors could be used for mitochondrial DNA. And if you go to DNA Painter, they have a tool there. You can upload or create your family tree, and then you can show the mitochondrial DNA path. And that'll help you visualize the ancestors from whom you inherited mitochondrial DNA. Now, you might want to seek out people that can be test takers for each of your different lines that you don't yourself carry the mitochondrial DNA for. And then that will help you to do further research and analysis. So one thing you can do, if you do take the full sequence test at Family Tree DNA, you can contact your matches and see if you can try to identify a common ancestor, but it can be kind of difficult to do that. But you do get a match list at Family Tree DNA, and at 23andMe you can use that haplogroup to see if you can make some connections, and the same with Living DNA. Now, Genetic Affairs does have an auto tree analysis of family trees for mitochondrial or Y-DNA matches, and so that can help you to do some more analysis. And then just know that Family Tree DNA Advanced Matches is the only tool that uses all types of DNA available to help you determine how closely related a match may be. So Family Tree DNA is a great place to do your testing, the Y-DNA and mitochondrial DNA, and also the autosomal, so you have all that together. Now, if you want to learn other resources, the iZog Wiki is a great place to start. It brings together a lot of different articles. And there are a lot of projects out there for mitochondrial DNA that are not necessarily about genealogy, but, these geographical projects or the phyllo tree, trying to see where the earliest ancestor came from. I know that on Living DNA, my son tested there, so he got the haplogroup for his mitochondrial DNA. He inherited that from me, so it's my mitochondrial line, and it goes back to Scandinavia, to the Sami people, Saami. So that was fascinating to me because that is my maternal line, straight back to Denmark. So that was kind of fun to see that actually show up in his DNA as his mitochondrial line. So if you're interested in any of those projects, you can certainly start exploring that and see how you can use that in your genealogy.
Let's talk about Y-DNA and some tools to help with that. Of course, like you mentioned, there's the DNA Painter ancestral trees that shows the mito and Y-DNA inheritance path, which is pretty easy. It's just father's father's father. Then there's also the Y-DNA match list at Family Tree DNA, which is the only company that actually gives you lists of Y-DNA matches. There's some other places you can find Y-DNA matches, like the mitoydna.org database. And so if people have uploaded their Y results there, Diana has typed in the results from her Royston project that was done by Sorensen DNA there. So some people have uploaded or added their results there, especially for these defunct companies. You can also look at yfull.com for more Y-DNA matching, and ydnawarehouse.org. So those are some different third-party databases for Y-DNA matches, but the only testing company that gives you a match list is Family Tree DNA, Y-DNA tests. And so that's where you would start. Now, within some of the companies who provide a Y-DNA haplogroup, you can learn more about the haplogroup predictions. So Family Tree DNA will give you haplogroups, and 23andMe, Living DNA, and GEDmatch can all provide haplogroups. The surname and haplogroup projects at Family Tree DNA are very useful. These projects help you identify which markers differ between you and those of a match. And then those differences can be used to identify significant mutations and help you identify other individuals who do not match. And then their results can be negative evidence for descent from a certain ancestor, which is often very useful. So, those surname projects and other projects like haplogroup projects can be extremely, extremely useful. And I'm a member of several surname projects with various test takers. So, the family tree DNA Y matches also has a tool called the TIP calculator. This is a time predictor and it estimates the number of generations between you and your Y DNA match and the common ancestor. When we came out with our book, Research Like a Pro with DNA, the TIP report was kind of outdated. And very recently, Family Tree DNA has released a new TIP report, which has used a new scientific algorithm to help calculate the time to the common ancestor. So be sure to check it out if you haven't seen it recently. The TIP report is great now. It's much better. It provides hopefully more accurate year ranges for the time when the common ancestor lived. There's a tool at the Family Tree DNA website called the YDNA haplotree, which can help you find out where you are in the tree. And they've recently updated this quite a bit too to make it better. And there's been a lot of updates at Family Tree DNA with all of their YDNA tools. I encourage you to check out the Family Tree DNA blog to see these recent updates and additions. And there's just a lot of new wonderful tools available there.
Right. Y-DNA is exciting, isn't it? How they continue to get better with the analysis and giving us more tools, especially with the big Y700 tests. It's amazing. Well, lastly, let's talk just a little bit about XDNA. And we really need some tools to help us visualize XDNA inheritance in the family tree. And so we want to make sure we understand that. And just like we could with the mitochondrial DNA, we can go to DNA Painter. And if we've uploaded or created a family tree there, then we can use the DNA filters tool to show us all the ancestors that potentially passed XDNA to us. And then when we're looking at XDNA matches on the websites that provide that, we can figure out that it can only be this certain subgroup of our ancestors who we matched with on that X chromosome. So charting is really helpful, and I know I have a big pedigree chart here up on my wall, and I've gone through and just marked people that I inherited X from I actually use Blaine Bettinger's XDNA charts. Those are on his website, and it clearly shows it. And once you understand the inheritance, you can just pretty much do it yourself, but it's nice to have a chart to look at and make sure you're marking correctly which ancestors gave you the X chromosome. Debbie Parker Wayne has fillable XDNA charts. Those are on debbiewayne.com. There are also charts through the Progeny charting software. And JMC Turner has fillable XDNA charts. So you can do some different things, different tools out there to help you see that. And once you have that figured out, then you can go to town trying to figure out how you match up with your XDNA matches. And then you can also compare your list of ancestors who possibly passed that XDNA on with your matches. And some tools that will help you there are the genealogical DNA analysis tool or WikiTree. So those are some interesting tools that I haven't tried, but there's so many tools. I have used WikiTree before, and that's a great tree. It's a collaborative tree that lets you put DNA evidence on, and so you can put your haplogroups on there and start comparing with people using WikiTree, which is great. And then finally, we can do some biogeographical information for the X chromosome. So we commonly call that ethnicity estimates. And so we can look to see the locations that our ancestors belonged at some point in the past on the X chromosome, and we get that through 23andMe because they show you your ancestry composition on each chromosome, and so that could really help as you are trying to figure out how you connect with a DNA match to see what ethnicity or biogeographical information that X chromosome reports. So some fun things that maybe you hadn't thought of doing before.
That's great. It really makes me want to dig into XDNA matches. And every time that I have done that, it's been fascinating because I love how in the XDNA inheritance pattern, if you go through a line where it goes female, male, that X chromosome is being passed down intact from the male to the female. So it's like preserving an older chromosome and segments from that generation because it's not getting recombined when it's passed from the male. The full X chromosome is passed down to the daughter. So it's exciting to find matches that go through that pattern because it can be for a more distant ancestor. Now let's talk about your task. At the end of each chapter in Research Like a Pro with DNA, we have a task for you to do. And so after learning about all these different tools, you need to then try using some of them. We encourage you to practice trying out a tool that piqued your interest, something that might help you in your research that you haven't tried before, and think about the answers to these two questions. Why should I use this DNA tool? How can it help me with your current project you're working on? Sometimes we need to just learn more about the type of DNA that we are using and understanding that better. So sometimes this chapter, the task is just to learn and understand more about what's available and what you can do with it. And then hopefully that helps you with the next step of Research Like a Pro with DNA, which is research planning. And understanding what tools can help you might cause you to plan to use them.
That's exactly right. And I always like to compare our DNA research to our documentary research. So you might not be using any type of a land or a tax record in your documentary work because you just don't know how to do that. But it could be so useful. So it's the same thing with our DNA work. We might be just ignoring Y-DNA or XDNA or chromosome mapping. But when we dig into it, then we realize how we could use it. So we often just have to start and learn more about it and then see how it can help us. So thanks everyone for listening. We hope you have some fun this week experimenting with DNA tools, and we'll talk to you next time.
Okay, bye.
Bye-bye.
Thank you for listening. We hope that something you heard today will help you make progress in your research. If you want to learn more, purchase our books, Research Like a Pro and Research Like a Pro with DNA on Amazon.com and other booksellers. You can also register for our online courses or study groups of the same names. Learn more at familylocket.com/services. To share your progress and ask questions, join our private Facebook group by sending us your book receipt or joining our courses. To get updates in your email inbox each Monday, subscribe to our newsletter at familylocket.com/newsletter. Please subscribe, rate, and review our podcast. We read each review and are so thankful for them. We hope you'll start now to Research Like a Pro.